Okay, Assalamu alaikum warahmatullahi wabarakatuh, everyone. I'm AbdulRahman Farahat. I'm a third-year medical student, and today I'm delighted to be giving you the lecture, Regulation of Gene Expression. Now, before we start, here is my number and my email. Please feel free to text me whenever you want. And let's begin. Okay, so first of all, starting with differential gene expression. So differential gene expression basically tells you, actually, let me ask you right now, do you guys think every single cell in the body has a different genome or the same exact genome?
Same
Different.
genome.
Could you repeat that? I couldn't hear you. Same, yeah. So the same genome. So every single cell in the body, whether it's a hair cell, it's a skin cell, an eye cell, a GI cell, whatever it is, all the cells have the same exact genome. But what differentiates which cell becomes which is dependent on your gene expression or which proteins and genomes get expressed. And so taking a look at this example over here, you could see two different cells. They have the exact same genome, huh? But one cell decides to express this part of it. And this gives you, for example, a muscle cell. Another cell expresses the other end of the genome and it gives you a nerve cell. And this is how the body works. This is how all the cells work. This is the basic concept of differential gene expression. It basically tells you that all cells contain the same genetic material. So all the cells in the body have the same exact genetic material. That's what tells us which cells have or become what is dependent on gene expression. So differential gene expression, again, it basically tells you that all the cells contain the same genetic material. However, depending on which gene gets expressed, gives you the cell specialization. Is this concept clear with everyone? We could move on, huh? Okay. Now let's start from the basic structure of genetic material. So, of course, you guys know about the DNA, huh? Like, how does the DNA get packaged? How does it get stored? How does it come all together? This depends on those cute structures we have over here, starting with the histone proteins. So H2A, H2B, H3, H4, they are all histones. Hello. What do we need to know about those guys? That, let's say, therefore, right now, we want to duplicate them. And then we put all 8 together. So it looks something like this. This is called an octameric disk. Like, what does the octameric disk do? You have this octameric disk, and all the DNA and genetic material keeps on wrapping around that octameric disk. And so basically, what does this form? It forms a nucleosome. And it keeps on wrapping and packaging and tightening over each other until you actually have a full chromosome. Okay, now this is the basic structure of genetic material. Now we have types of genetic material. We have the euchromatin and heterochromatin. So before we actually read them, I gave you a glimpse about it. So could anybody really quickly tell me what's the difference between euchromatin and the heterochromatin? Okay, so euchromatin is loosely packed. And what does this lead to? If it's loosely packed, what would happen? Try to link it. Okay, hetero is slightly packed. Makes sense. So yeah, if it's loosely packed, it becomes more active. It's easily accessible to the whatever RNA polymerases, and so you could easily express it. Compared to heterochromatin, which is, like you said, it is tightly packed, and so it becomes inactive. Perfect. Amazing. Right. More stuff about them is that since it is loosely packed, they are not all condensed over each other. When you look at them under the microscope, they look lighter in color compared to heterochromatin, which very compressed over each other, appear darker. And finally, euchromatin is present in both prokaryotes and eukaryotes. Wow, amazing line. And heterochromatin is only present in eukaryotes. Hell. Now, what's the basic function of heterochromatin? No need to focus about it. It's generally there for the chromosomal stability. Okay, that's heterochromatin. Now, heterochromatin is also divided into 2 parts: facultative and constitutive heterochromatin. Okay, so like we wrote here, facultative is reversible and constitutive is irreversible. So we're basically telling you, in heterochromatin, if it is facultative, there is a chance that we could reverse it back to euchromatin and it expresses its gene, compared to constitutive, which is completely irreversible. Okay, perfect. Does this make sense with everyone? Are we all on the same page? If I'm going too quick, you could stop me anytime. I'll repeat over and over and over again. Okay. Now, moving on to something we call the transcriptional control of gene expression, specifically in prokaryote. So how we control transcription in prokaryotic cells, like bacteria. Now, the most common example for this would be the lac-A gene or lac-A operon. It's called the lac operon. So as you can see over here, it has a promoter region where you have the binding of the RNA polymerase. It starts its transcription. And then you have the operator region. Okay, so what's an operator? Basically, it's a DNA sequence that allows other DNA binding proteins or binding proteins in general to bind to it and either promote the gene expression or inhibit the gene expression. And then you have the LAC-Z, LAC-Y, LAC-A. These are all generally other enzymes that are used to break down lactose. So in the presence of lactose, if you could imagine with me, what happens is lactose is a sugar. It's like a sugar molecule like this. Imagine that lactose is in an environment. Imagine there is a prokaryotic cell, a bacteria, مثلا, in your stomach, and you drink milk. What happens? Milk contains a lot of lactose, by the way. But lactose meets with the bacteria, and this lactose comes to the operator region over here. It binds to it and it activates it. Why? It activates this operator region, promotes transcription, and basically you're getting all those enzymes that break down lactose. And that allows the bacteria, specifically E. coli, to actually feed on the lactose and gain energy out of it. But this is, in short, how a transcription is controlled. The gene expression is controlled in prokaryotes. Is this example clear with everyone? Can you repeat what is promoter and how is this different? Promoter is a place where the RNA polymerase binds to. However, it needs to be activated, and for it to continue, it needs to pass by an operator. Now, this operator is a sequence of DNA where you could have multiple other proteins that bind to it. But it's not like an RNA polymerase. It's always open to bind to this region. It will always, whether you have an RNA polymerase or not, it's always going to bind to the promoter region. However, it won't continue through if there's something blocking it here, which is like a regulatory protein. Okay, let's erase this again. Okay, let's repeat. So, okay, so one got it, the other. Okay, so let's repeat it really quickly again. So you have an RNA polymerase that would always bind to the promoter region. This is fixed. This would never stop. You'd always have an RNA polymerase that binds, but sometimes you don't need those enzymes. So how do you inhibit those enzymes? You get something like a regulatory protein, a certain repressor or an activator. It binds to the operator region, and it prevents the RNA polymerase from continuing and expressing those genes. But this way, you're controlling the amount of enzymes released. But for example, sometimes the E. coli would need its lactose to store. It doesn't need to break down all that lactose. But how does it control it? It needs to prevent enzyme production. And how does it prevent enzyme production? It needs to prevent the operator region. It cannot inhibit the promoter. The operator should be inhibited. Does this make sense? Hopefully. Okay. I'm hoping it did. If it didn't, you could tell me after. Okay. Now, moving on, we have two types of genome. We have the polycistronic and monocistronic. Could you guys tell me what's the difference between a polycistronic and monocistronic? From its name, you could kind of guess. Polycistronic, you'd have multiple genes and one promoter. Okay, perfect. From its name, polycistronic. Hello. And monocistronic would be a single gene by a single promoter. Makes sense. But Polycistronic, imagine this region would be the promoter, and it controls gene A, gene B, gene C, gene D, and gene E. All those genes under one promoter. Monocistronic from mono, one. So you'd have a single promoter, and you'd have this whole thing, which is a gene. You see exons and transexons. This is still the same gene, by the way. Okay, another question. Which one is found in prokaryote? Which one is found in eukaryotes? Polycistronic, yeah, polycistronic. Is it prokaryote or eukaryote? Pro or eu? Polypro, perfect. And mono, hello. Poly in prokaryote, perfect, perfect. Okay, so this is basically it. Prokaryotes have polycistronic genes, and eukaryotes have monocistronic. Amazing, amazing. Hello. Now, moving on to the actual depth of the session itself, transcriptional control. But for transcriptional control, you have two types. You have the structural chemical modifications, and you have the DNA binding protein. Structural chemical modifications, we have two types. You have to think about DNA methylation or demethylation. If there is methylation, then there could be the opposite. And you could also need to think about histone modifications, because you remember when we spoke about histones and the DNA wrapping around it. But if you control the histones, you could control whether the DNA is loose or tightly packed. Huh? Okay. Moving on, we also have the DNA binding proteins, and those we'll speak about later, inshallah. So they're basically gene proteins that, like I wrote over here, toggle on and off gene expression. Okay, moving on really quickly, starting with DNA methylation. Okay, starting with DNA methylation or demethylation, or specifically on CpG ions. Okay, so let's start with this part over here. What is this? Did anybody tell me? No. This is from a previous lecture. Let's test your knowledge. Data box. Okay, so what's the data box? It's basically a promoter, صح? A promoter region. حلو. Okay, and after the data box, what do we have? We have C, G, C, G. I think in the other file I said cysteine instead of cytosine. Yeah, I'm sorry about that. It's cytosine. So you have cytosine or C beside guanosine, and then you have a phosphodiesterase bond here, and then a G beside a C, another phosphodiesterase bond. So it's called a C phosphodiesterase G, CpG islands. But these are CpG islands. Now, let's talk about DNA methylation and demethylation. So let's say we add a methyl group to the C. You add methyl to the C, MC. So what should happen? Would it activate or inhibit gene expression? What do you guys think? If you think it's activate, it inhibits. Okay, we have inhibit and activate. Yalla. Who else? More answers? Okay. So CpG, when you add a methyl group to the C, it actually inhibits methylation of DNA. DNA methylation inhibits gene expression. So as we said here, methylation of cytosine, not cysteine, turns gene expression off. By the enzyme, you could get it, DNA, because it works on DNA, methyl, you're adding a methyl group, transferase. You're transferring that methyl to the cytosine. Okay. So if you demethylate it or you remove the methyl group, you could get the opposite. So you're activating it, and the enzyme is 1011 translocase. I watched the recording, and the doctor sort of spoke about the enzymes really quickly. Back in our days, we didn't really have to know the enzymes, but you guys could try to memorize them really quickly. If DNA methyl transferase, add methyl group, and 1011 translocase. Okay, moving on. This is DNA methylation, demethylation. Pretty simple, صح? Could you move on to the post-translation and histone modification? This session is going to be, it's already going really quickly, alhamdulillah. Okay, I think we could move on to post-translational histone modification. Okay, so we have a lot. We have histone acetylation, histone methylation, histone phosphorylation. It's a lot of boring stuff, I know. But we're going to go through them really quickly. And I think the one the doctor really focused on was histone methylation. So keep that in mind at least. Now, for starting with histone acetylation. From its name, acetylation, it actually, I showed it to you guys. Acetylation activates. So it basically, activates the transcription and promotes gene expression. While removing the acetyl group would repress gene expression. طبعا, which enzyme? I doubt, to be honest, and you guys would be asked about this, but remember it just in case. If it comes in an MCQ, you could easily grasp on it. So acetylation typically happens on lysine, AL. acetylation activates lysine, and then you have activation of gene. Now, when it comes to the enzymes, again, it works on histone, so it's called histone. You're transferring an acetyl group, the acetyl transferase, and it activates transcription. The acetylation activates. Hell, histone deacetylation, you're removing the acetylases, remove the acetyl group from the histone protein, and so it represses transcription. Does this one make sense? Moving on. Hell. Histone methylation. Okay, so methylation, M for mixed effects. What do you mean by mixed effect? I mean, whether you add methyl histone, remember, don't confuse between them. Adding a methyl group to DNA on the CPG island would cause. Well, I want the whole chat to say something the same word, please. Inhibition. Okay. Anyone else? Anyone to challenge? Okay. Inhibition. Perfect. Yes. Yes. More, please. Yes. I like this. Yeah, adding a methyl group to DNA inhibits, while adding a methyl group to histone could either activate or repress. But don't confuse them. Histone is completely different from DNA. Hell, and this is why I wrote M for mixed effects. So it could either activate or repress. Again, the enzymes would make sense. Adding you're adding a methyl group to histone. That starts with histone. You're transferring methyl transferase, and histone demethylated. Which amino acids? Lysine and arginine. Mumtaz, are we all good? Moving on. Histone phosphorylation. For histone phosphorylation, okay, let's see who of you watched the other part session, protein synthesis. So, phosphorylation. What are the enzymes for phosphorylation? Which enzyme adds phosphate? Please, more than 5 people. Yes, 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 yes. Kinases. And which enzymes remove phosphate? Phosphatases. Mumtaz. So this is the whole idea. And no, histone phosphorylation, you have two enzymes, kinases and phosphatases. Kinases add phosphate groups, and phosphatases remove phosphate groups. Again, it could either activate or repress transcription. Mumtaz. Which enzymes? Serine, threonine, and tyrosine. I tried so hard to look for a mnemonic for this, but it's so difficult. But you could try to remember ST and all the T's. But S, serine, and then you have only 2 T amino acids. Okay, no, I actually have 3, but 2 of them, threonine and tyrosine, not tryptophan. Don't confuse. Moving on. Finally, ubiquitination. Try to remember this forever, and I'm pretty sure you guys took a lecture about this, hopefully. Ubiquitination basically means you're assigning that protein to go die. You're degrading it. But once a protein gets ubiquitinated, it goes for degradation, whatever that protein is. So histone is a protein. But when you say histone ubiquitination, it means you're adding a ubiquitin to that histone. And so what happens after that? It goes for degradation. And then de-ubiquitinases remove ubiquitin. Mumtaz. Moving on. Now, okay. So we're also done with this part over here. Hell, are we all clear to this point right now? Does anybody want me to repeat anything at all? Is the lecture easy? Okay, so I'm getting nothing, but I hope that's a good sign, hopefully. Moving on. DNA binding proteins. Can you please repeat the histone acetylation? Okay, so for histone acetylation, remember A for activation. But remember, once you add an acetyl group, remember A for activation. Adding an acetyl group to the histone protein makes it looser, makes the genome looser or less tightly packed, and this activates transcription. While removing an acetyl group, you're removing an A, you're removing the activation, and so it becomes more tightly packed, and this will inhibit your transcription. Which amino acid? Lysine. Try to remember. I try to remember it with all. Is the amino acid where the modification happens on the DNA? No, it's on histone, and histones are proteins. And what are proteins made of? They're made of amino acids. You're adding an acetyl group or a methyl group or whatever modification it is on that amino acid, on the histone, not the DNA. Again, remember, histone modification, not DNA modification. Makes sense? Okay. Alhamdulillah. Moving on. Now, for DNA binding proteins. For DNA binding proteins, it's so confusing in the lecture, and even I got confused with it. But I'm going to give you a mathematical equation to memorize it, inshallah, hopefully. But first of all, let's classify. So added on the amino acid of the histone. Yes, perfect. So histones are protein. Yeah, perfect. Histones are made up of amino acids, and that specific amino acid, it have the addition of acetylation or yes, exactly. Okay, so coming back to DNA binding proteins, I need you guys to remember two things. You have cis-acting elements and trans-acting elements. The cis-acting elements, basically, cis means, did you take the cis and trans side of the Golgi apparatus as well? So cis means on the same or close by, near. The cis regulatory elements are basically DNA sequence. So they are sequences like any other DNA, يعني genome. فمثلا it could be an ACTG, whatever it is. It's a DNA sequence located on the same chromosome, and it controls whether this genome gets activated or repressed. Same thing with the trans-regulatory elements. So you actually no, not the same. Trans-regulatory elements, from their name, trans. So they are made somewhere away from the chromosome itself, and they act on a different chromosome. Right? So I have chromosome 1 and chromosome 2. So they are made, they are synthesized on chromosome 1, and they go act on chromosome 2. This is how trans-regulatory elements work. Now, for cis-regulatory elements, they are found in chromosome 2, and they act on chromosome 2, both of them. Makes sense? What are the examples we have? For cis-regulatory elements, we have the promoter, the enhancer, the silencer, and the insulator. A lot of terms. I know it's so boring, but we'll go through them really quickly now, and inshallah, by the end of this, you'll have it easily memorized. And for the trans-regulatory elements, you divide them into proteins and RNAs. For proteins, you have activators, repressors, mediators. These I'm basically listing names, but now we're going to actually use them. Let's start with this. This is what's written in the lecture, and it completely confused me. I don't know if I'm dumb, but I wasn't able to take it easily. But it says gene control is mediated by transacting factors, activators, and repressors binding to cis-acting. It's so long that it confuses me, but I turned it into this, a simple equation. You have gene control is basically transacting factors plus cis-acting elements. When they come together, you're deciding whether this gene should be expressed or inhibited. Perfect. So now let's put them together. So remember, positives always love each other. But activator is a positive, and an enhancer is a positive, and they always love each other. An activator and an enhancer would come together, and since they are positive, what would they do? They would activate the gene expression. While negative people, they sit beside each other because they are all negative. They all hate each other. But you have a repressor, which is a negative, and a silencer, which is also a negative, and they all work together to what? Inhibit stuff. They just spread their negativity around people. But this is the whole concept. Now, we said we have enhancers, silencers, promoters, and if you remember, we said insulators. What are insulators? Insulators are completely the opposite. They come and they're that annoying kid that keeps on switching everything around. But enhancers, or let's see this, activator plus an enhancer and an insulator would inhibit the gene expression. That does the opposite. And the repressor would always go with the silencer. But when you add an insulator, it would activate the gene expression. I hope this part makes sense. Because this part was actually one of the toughest things for me personally when I was studying it. I hope it did make sense. If it didn't, tell me. I could repeat again and again. Yes, it did. Alhamdulillah. Okay, let's repeat. Again, I'll repeat from this. Let's erase. Okay, so this is what your slide has. Try to remember, if you remember, okay, let's actually start from this part. You have cis-acting elements and trans-acting. Cis-acting act on the, okay, you guys help me with this. Cis-acting help on the same chromosome. Trans-regulatory, you better be saying this with me, Sarah. Trans-acting act on different chromosomes. So they could be synthesized in chromosome number 1, and they go act on chromosome number 2 or 3 or 4 or 5. Okay? This is how trans-regulatory elements work. What are the examples we have? For cis-regulatory, we have promoter, we have an enhancer, we have a silencer, and we have an insulator. For trans-acting, we have the proteins and the RNAs. The proteins, you have activators, repressors, and mediators. So proteins typically work with these acting elements. But always remember, activators love enhancers. So activators and enhancers always come together. Repressors and silencers also always come together. And an insulator, you keep them for the end. They are alone. Hell. But let's put them all together here. So once an activator meets with an enhancer, but imagine a genome like this. Here you have an enhancer. This guy is an enhancer. Hell. And you have this protein going around. This guy is an activator. This activator goes and binds to the enhancer. What happens after that? It allows the RNA polymerase to continue transcribing the gene, but activates gene expression. Now, for repressors and silencers, again, you'd have this genome over here. You'd have a repressor, and then not a repressor, a silencer. This is a silencer, and you have the repressor binding to the silencer. What would happen? You'd inhibit gene expression. A repressor and a silencer, they bind together and they inhibit gene expression. Okay. And now for insulators. Okay, for insulators, you'd have activator and an enhancer. And then once you add an insulator, it does the complete opposite. So we said activator and enhancer would typically activate your gene expression, activator and enhancer. But then once you add an insulator, it does the complete opposite. It inhibits gene expression. While repressor and silencer, again, we said they inhibit gene expression. But once you add an insulator, it activates gene expression. Make sense? Hopefully. It's fine. If you didn't get it again, don't worry. You could say, okay, do we have both on the same gene at the same time? Yes. So, مثلا, like this example over here, you see, مثلا, because you have to think of an enhancer as a DNA sequence. If you remember, we said this. An enhancer is a DNA sequence. So you'd have this DNA sequence, and an enhancer would always be there. While a transregulatory element is either an already made protein or an RNA. But it's a protein that's made. It's transacting. It's not a DNA sequence. And it binds to that DNA sequence. And this is where they actually both come on the same gene. Hello? Yes, okay, alhamdulillah. Okay, do mediators have no effects? Mediators wasn't spoken about in the lectures, but as long as it wasn't spoken about, forget about it. No need. I just wrote it there for the sake of completion because it was there, but it wasn't spoken. Cool. Perfect. Plus, we're all on the same page. Yalla. Now, for activators, okay, this part might get a little confusing, but inshallah we'll make it easy. Okay, so for activators, we have two domains. So imagine it's going to look something like this. This is how the protein looks like this two-legged thing. You'd have a domain that binds to the DNA over here, and then a domain that adds whatever enzymes are ready or whatever that activates the transcription and makes this part activate. So you have two domains, one that binds and the other that actually causes the activation. Okay? And how does it activate it? It facilitates or attracts more proteins to come and attach to the gene. So I could attract an RNA polymerase, come here, you bind, you go to transcription or whatever. Hello? So those are the two domains that are activated. One that binds DNA and another that activates transcription. Hello. Now, we also spoke about domains and motives. And this is how I like to remember domains and motives. Honestly, this is what's mentioned, and this is what the doctor wants you to know. And you could remember this. This is how I like to understand. But really quickly, what you need to know, a motif is a very short amino acid sequence. So it's a protein. It's a very short protein that won't be able to do a function on its own. And following the doctor's words, he said, if you have this protein and you throw it in the cell, it won't have a function on its own. It's basically useless. However, a domain is a long amino acid sequence that can do a function on its own. And this is how I like to see it. Like a motor is this hand over here. On its own, it won't be able to reach anything. All it does is grabs, but it won't be able to reach anything. It's a motive. It's very useless to me right now. But once I get a complete arm and hand and all this is my domain. This could reach and grab. I could do two functions right now. So does this make sense? A motif on its own is basically useless and it's very short. So like this hand, it's very small, while a domain is a long amino acid sequence that can do a function on its own. If you could remember this part, that's all you need to know, to be honest, about it. حلو. Okay, we're basically done with the lecture, صراحة, alhamdulillah. But really quickly, gluconeogenesis. I'm going to ask you guys some questions, and you could tell me what they are. First, what is gluconeogenesis? Making glucose, production of glucose. Perfect. Yeah, so we're producing glucose. حلو, ممتاز. Now, why do you need gluconeogenesis? When do we need gluconeogenesis? In what situation do you think we need glucose? When fasting. Perfect. Okay. Any other situation when no glucose in the body? حلو. Starvation. Okay. Okay, that's all around the same area. There's another situation where you'd make a lot more glucose. So when you're fasting, when you're in stress. So perfect. So think of it this way. It's when you're using too much glucose or when you have no glucose to begin with. So when you have no glucose to begin with is when you're fasting. And when you're using too much glucose, that's during periods of stress. Like, what are the hormones you need to remember? During starvation, what gets released to help make glucose? Glucagon. Okay, and during stress? Cortisol. Perfect. Okay, now basically that's like simple stuff here and there you could easily get asked on. You guys know your stuff really well. Alhamdulillah. Moving on. Now let's take a look at this diagram. It's the exact same one in your lectures, but because I liked it, it looked really good. Starting with glucagon. Glucagon would come and bind to a membrane-associated receptor. But remember this. This is actually a point to remember. Glucagon cannot cross the membrane on its own. It binds to a receptor on the membrane. And this increases cyclic AMP. It activates protein kinase A. Again, kinases the add phosphates. And this activates CREB, cyclic AMP response element binding protein. You don't need to know the name. It's cyclic AMP response element binding protein. And what does this guy do? Once this guy gets activated, as you can see, it goes to the DNA. This is DNA, by the way. Goes to the DNA and it binds to its element over here, the cyclic AMP response element. Okay? And once this binding happens, it causes activation of this gene over here, PepCK gene. PepCK is phosphoenol pyruvate carboxykinase. You don't even need to know about the name. All you need to know is that this gene becomes messenger RNA, it becomes a protein, and this enzyme is the enzyme you need. All you need to know is this enzyme is the enzyme you need to make glucose. Without this enzyme, you won't be able to make glucose. But the whole purpose of this cycle is you're trying to increase this enzyme so that you're increasing the production of glucose. Hell, does this pathway make sense? This is when you're fasting. When you're fasting, you have increased. You could get easily asked about this in MCQs. I'll give you an example. You could have a whole long case about some guy during Ramadan, he's fasting, and they're asking you which process increases glucose during his fasting. And then you'd have answer choices. One of the answer choices would be high cortisol or whatever that would go to the DNA and bind to GRE or whatever. This is the wrong answer choice. You need to have an answer choice that works on this pathway over here. Okay? But take those small consideration into mind. Okay. But does this glucagon pathway make sense? Could you go to the cortisol pathway? Yes. Yes. Yeah. Can you please repeat it? Okay. Let's delete. Okay. Starting with glucagon. Glucagon comes and binds to this receptor over here. It's a membrane-associated receptor. It's a receptor on the membrane. And what does this do? It increases your intracellular cyclic AMP. It's a whole process that you don't need to worry about. You just need to remember that when glucagon binds to that receptor, it increases cyclic AMP. And cyclic AMP, always remember this actually, this is a typical thing. Cyclic AMP always goes to a protein kinase A. Hell, always goes to a protein kinase A. And protein kinase A adds a phosphate to what? to this guy. It's called CREB. Forget about the other name. It's called CREB. So it adds the phosphate to CREB. Once it adds the phosphate to CREB, it's basically telling CREB, you're activated, yalla, go do your job. What does CREB do? It goes, gets activated, and it binds to its response element on the DNA over here. So it enhances the transcription of messenger RNA. This is basically, you could guess of this as an activator, and this is the enhancer. This is an enhancer, and this is the activator. And once it binds to it, as we said, activator plus enhancer would activate gene transcription. And so this activation would activate this gene transcription of PepCK gene. What is PepCK? It's basically the enzyme, the main enzyme you need to make glucose. Hello? Yes, thank you. Okay. Now, moving on to the cortisol pathway. For the cortisol pathway, again, differentiate between some. Cortisol, unlike glucagon, could cross the membrane. So, what was the question? I'm trying to remember. The doctor asked you guys, I forgot. Anyways, but cortisol, again, let's start with the back. Cortisol crosses the membrane, the cell membrane, and it binds to an intracellular receptor, unlike glucagon, which is that which one activator and which one enhancer in glucagon side. The enhancer, if you remember, we said, why would cortisol pathway be a wrong answer for a fasting person? Because you're not in period of stress. Cortisol would increase in periods of stress, while glucagon increases in periods of fast. Always remember that, right? Unless you're stressed during fasting, then yeah, you could argue with that, Sarah. But other than that, yeah. Okay, now coming back to which one activator and which one enhancer. Okay, an enhancer, if you remember, we said it's a cis-acting element, and so it's a DNA sequence. So this is an enhancer. If you look at this image, CRE would be your enhancer, huh? Because it's a DNA binding, it's the DNA sequence. While the activator is CREB, CREB. Why? Because it comes from outside and it goes inside and it binds. It's a protein that binds to the CRE. Does this make sense? Okay, perfect. Alhamdulillah. Cortisol. Coming back to cortisol. Cortisol. Yeah, I remember the question. Thank you for asking. Which organ does cortisol get secreted from? Adrenal medulla. Yes, good. So cortisol gets released during periods of stress. It crosses the blood-brain, it crosses the cell membrane, and it binds to an intracellular receptor. There is no receptor for cholesterol, for cortisol outside. It goes inside and binds to an intracellular receptor. And then cortisol goes through and it forms this complex. This is a complex, by the way. It's cortisol plus its intracellular receptor. And this is basically your activator complex, you could say. And what does the activator complex do? Again, it goes inside, it binds to its response element or your enhancer region, GRE, glucocorticoid response element. And what does this do? Again, the exact same thing. It goes, it activates PepCK. And what is PepCK? Your main enzyme for glucose synthesis. Okay, we're done with this pathway. Hopefully you all got this. خلاص. If the doctor asks you any question about this, then خلاص, يعني you guys should have it on mind and you could imagine the whole pathway and it's easy for you, inshallah. Huh? Could we move on? Anybody have an issue? Okay, now. No, okay. So coming back to this division over here. So how do I delete? Okay, erase pen. So first we spoke about this. So we're done with this part. And we spoke about this and we gave its example. Now we're speaking about RNAs. How do RNAs control your gene expression? But this is what we call something. It's called RNA interference. And there are two main RNAs that do this, microRNA and small interfering RNA. Okay? Okay, how do we differentiate between them? This is, again, a table from your side. It basically tells you everything you need to know for now. But small interfering RNA, it comes or synthesized from double-stranded RNA, as we're going to talk in this image right now. While microRNA from single strand. Again, we'll explain this. Don't worry. I'm just saying stuff right now. Small interfering RNA is processed by an enzyme called dicer in the cytoplasm. Dicer is in the cytoplasm, while DROSHA is processed by two enzymes. While microRNA is processed by two enzymes, DROSHA and dicer. DROSHA is in the nucleus and dicer in the cytoplasm. Again, we'll explain it. Now, differentiate between them. Single small interfering RNAs target a single messenger RNA. S with an S, a single with a single. Okay, a microRNA targets multiple, an M with a multiple. Hell. Now, if it's targeting a single messenger RNA, you should think, it should be fully complementary to that messenger RNA, to a single specific messenger RNA. While since this guy binds to multiple RNAs, it could partially be complementary. It sometimes binds it and sometimes not. Hell, this table, really quickly, we could look over this. Okay, but this one is specifically for the small interfering RNAs. But let's start from the beginning over here because it kind of gets confusing. This guy, we said that if we go back to this table, it says that it comes from double-stranded RNA. But this is your double-stranded RNA. This is a double-stranded RNA, and this is a double-stranded RNA. And they form a loop like this. Okay? This guy goes to the cytoplasm. In the cytoplasm, what do you guys have? What enzyme in the cytoplasm? Dicer or DROSA? DROSA. Hell, dicer. A dicer in the cytoplasm. What does it do? It cuts off this point, and it forms a small interfering RNA. Again, here is another example of long double-stranded RNA. Again, you have dicer, and the final route is transfection. Transfection is basically you're injecting it in or giving it to that person through another artificial way. Okay, you got your small interfering RNA. What happens next? Small interfering RNA goes and it forms a complex with this guy, argonaute proteins. Argonaute proteins. The small interfering RNA is the argonaute proteins. They come together. Perfect. They form a risk complex, RNA induced silencing complex. RNA induced silencing complex. Okay, what does it do next? You see those two pathways, but it sees this messenger RNA. It binds to it. And you have either one of two choices happen. Either it becomes an inexact sequence match. But you could see in the beginning here, there's a binding and then there is separation. But this is inexact. And so basically, this inhibits translation, prevents translation. The other scenario is where it perfectly matches with it. And once it perfectly matches, you have complete degradation. Hell, this is how small interfering RNAs work. I doubt that you'd get really asked about the specifics. That's all I need you guys to focus on would be dicer is the enzyme that processes the small interfering RNA, and it's found in the cytoplasm. This is one point. Second point, small interfering RNA react with argonaute proteins, and they give you risk complex. And that's about it. That's actually the 2 points that you need to know. So this is how the whole thing works. This is the pathway for small interfering RNA. For micro RNAs, it's the exact same thing. But the difference is, here we said it comes from double-strand. It's not small interfering comes from double-strand. But microRNAs come from single-strand. But you'd have normal gene transcription. You'd form a messenger RNA, which they call a pri-microRNA. And the pri-microRNA, what's found in the nucleus? What processes it? What cleaves it? DROSHA. Yeah. So DROSHA forms your pre-microRNA. But is DROSHA enough? No, it needs another guy. What's the other guy? Dicer. Where is he found? Cytoplasm. Pre-microRNA goes to the cytoplasm, and through the action of dicer, it cleaves it into a fully matured microRNA. Now, this microRNA would go on and do the same pathway, but it sees an argonaute protein, it forms a risk complex, and then it has the same actions on messenger RNA. It inhibits them. Okay, you're trying to, okay, I'll tell you now how, really quickly, microRNAs and small interfering RNAs, when they meet, they prevent the transcription. But we said here it causes degradation. So here, how does it inhibit translation? It prevents ribosome from binding because it's double-stranded. Ribosomes need a single-stranded messenger RNA. So when it sees the double-stranded, this is a useless messenger RNA to me now. I can't see the other complementary nucleotides. Huh? But ribosomes won't be able to bind. But this is how they prevent translation. But this is sort of post-translational modification or control of gene expression. This is how you're controlling. I hope I didn't say too much to confuse you guys here, but if I did, you could tell me right now, yeah. All good? Okay, we're basically done with the lecture. Do you want me to repeat the whole RNA interference pathway? Like the whole small interfering RNA and the microRNAs. You want me to repeat both? No, just the mechanism. Okay, so مثلا we said over here إنه the RISC complex is formed. It sees a messenger RNA like this. And you have two scenarios. One scenario is when it completely perfectly matches, you'd have complete degradation. صح? This is agreed upon. This is an easy one. صح? We could agree on this. We could forget about it. Could we move on to the next? Okay, we'll move on to the next. The other pathway is when it binds to it, but there is an inexact, here it's written inexact sequence match. But they don't all complete. If you see the pink line and the blue line, they bind at the beginning, and then at the end, they sort of don't bind together. So this is an inexact sequence match. And what happens? It basically prevents a ribosome from coming and binding to this region and reading the messenger RNA to give you an amino acid. But this is how it prevents translation. Make sense? Okay. Now, this slide, I'm really sorry, guys. You're gonna have to memorize للأسف. I tried really hard to make it simple, but unfortunately, I remember taking it we took it in MOL 2, and it's a terrible slide. You need to understand, because it could typically is the sense strand of the sRNA removed. Sense strand. It's not of the sRNA. A sense strand is a normal messenger RNA in the cytoplasm. It's any messenger RNA, basically. It's telling you it's any messenger RNA in the cytoplasm. And it binds, once it binds yani, with the RISC complex, like this complex over here, you see this is the normal messenger RNA. And this is the RISC complex with the SI. And it prevents any binding of ribosomes. Okay, if it's confusing you too much, you could forget about it because I don't remember the doctor mentioning that. I'm trying to give you a fuller picture. Okay. Now back to this techniques, try to memorize it as much as possible. عشان you could easily get a question مثلا, like, let me ask you right now. Which of the following would you use to measure the RNA levels? Let's see if you guys know. In situ? Nope. Not the any other guess. Yes, okay. So which of the following would you use for measuring RNA level? That would be the northern blot. حلو. You said in situ. In situ measures DNA or RNA within fixed stitches. It doesn't measure the specific. When the question specifically asks you about measuring the RNA level, then it's northern blot. If you're measuring مثلا specific messenger RNA, what would it be? Or if you're measuring, okay, two questions. If you're measuring specific messenger RNAs, and if you're measuring the whole RNA sequence in the body. There are two different techniques. Okay, PCR is for the specific. Yes, perfect. And which one for the whole mRNA sequence? Microarray. Okay. Oh, it's not microarray. It's for RNA sequencing. I got it wrong. Okay, RNA sequencing for the transcriptome. Transcriptome is basically the whole RNA in your body. It's called the transcriptome. Okay. Microarray measures thousands of genes simultaneously. So yeah, this part you could get simple MCQs on, and you could get acid. Okay, finally. So if you want to correlate it, everything we said before was basically transcription control. We're trying to control formation of messenger RNA, controlling the formation of messenger RNA. Now we want to control formation of a protein. And our last guy is called translational control. And we have an example. This is our example with ferritin. So you guys took ferritin. Ferritin is a protein found inside your cells and it binds iron. Okay, and how does this work? This is an RNA sequence, by the way. This is an RNA sequence, not a DNA sequence. And you have this region over here. Remember, at the beginning of an RNA sequence, you have the 5' untranslated region. Okay, so you have two scenarios. You have high iron and low iron. When you have a high iron concentration, do you think you need ferritin or no? So would you transcribe or translate ferritin or no? You didn't need in high concentration. You didn't need ferritin. You'd let iron stay in. Yeah. So we need. You will translate. Yes, exactly. Maybe I asked the question wrong. It's probably my fault. But when you have high concentration of iron, you can't let iron be around. I'll start translating ferritin to store that iron. So this is how it happens. Oops. So I have this guy called iron response element binding protein. IRE binding protein. Just remember it as that. And so it basically, I wrote it here like it's a repressor. So if you remember, it's a repressor. It binds to a silencer. And then the 5' untranslated region, it could be your silencer. So this is the IRE. Once the repressor binds to the silencer, it would inhibit the translation. Like, how do you control your repressor? When you have so much iron, the iron would bind to the repressor and it prevents it from binding to the silencer. And so this way, you're promoting or you're activating your translation. Hell, now on the other side, when you have low iron concentration, you don't have any iron to bind to the repressor. So the repressor is free and it wants to grab something. What does it grab? Your silence. And once it grabs its silencer, it blocks your translation and you have no more ferritin. And so you can no longer, you're not storing iron anymore, which is good. You don't want to store iron in low iron concentration. You need to provide the body with iron. Make sense? Yes. Alhamdulillah. Okay. On to our final concept of today, epigenetic inheritance. Is the silencer the 5' region? Yes and no, because look, I don't want you guys to remember it as silencer per se, because this is translational modification or control, not transcription. If it was a DNA sequence, I would confidently say, no, this is a silencer. Because a silencer is DNA sequence. However, since it's an RNA, not a DNA, you can't really confidently say it's a silencer. Okay, let's explain. Okay, type. So now you have two scenarios. You have a low iron scenario and the high iron. When you have low iron scenario, okay, let's go back to the high iron actually. For the high iron scenario, you'd have a lot of iron in the body. And this iron would grab onto this protein over here, which is called an iron response element binding protein. Iron response, which is basically a repressor in this case. Right? This guy goes and binds to the 5' untranslated region. Typically, it goes and binds there. However, since you have iron binding to it, but this guy doesn't need to bind to this region anymore. It has something bound to it, but doesn't do this stuff. And now your ribosome is free to attach to the 5' untranslated region and express or translate your ferritin protein. So you start having ferritin, and now you could store your iron when you have so much iron, high iron level. Now, in low iron concentrations, what happens is, remember this guy, the inactive or the iron response element binding protein, it needs iron. Now, in the low iron concentration, there is no iron, but it's free. It's free over here. And what it does is, it goes and binds to the 5' untranslated region. And once it binds there, it prevents the ribosomes from coming and translating this protein. And so you'd have no ferritin production. Hopefully it makes sense. Okay. Okay, epigenetic inheritance is our final concept for today. And all you need to know about epigenetic inheritance is that there are changes. They are changes in your gene expression, so different genes. However, it's due to something, any underlying cause other than mutation or any change in the DNA sequence itself. So you'd have the same sequence of DNA. You'd have A, C, G, whatever, but it never changes. And there's no mutations that happen. And you still have changes in your gene expression. Right? Some key points you could try to remember is one, there is no DNA changes. Epigenetic inheritance does not change the DNA code itself, but it affects how the genes are used. It affects how the genes are transcribed. Two, it is reversible. So unlike the mutations, which are irreversible, epigenetic changes can sometimes be reversed. Examples of epigenetic inheritances would be factors like diet, factors like stress, and toxins. These are changes that could affect your gene expression. So example of stress, مثلا, like we just gave this example over here, the glucagon pathway. In periods of stress, you don't have a change in your DNA sequence at all. Yes, you have a lot of cortisol, a lot of hormones. They go change the whole gene expression and they increase PEP-CK. Okay? So this is an example, but they don't change your DNA sequence itself, but they do change your expression. Make sense? And this could be okay. Hello, are we done? And we're done. Thank you so much, guys, for your time. If you do have any questions, please text me, and I'll be sending you guys, inshallah, the question back as soon as possible. Good luck in your MOL midterm, Tawfiq.